Good morning, this is Dr. Rajesh Yadav from Department of Biochemistry. Today we will discuss about the membrane lipid. So we will discuss about what is the membrane lipid and membrane fluid mosaic model and membrane lipid, role of cholesterol in the cell membrane, asymmetry of the membrane due to the composition of the lipids and movement of the lipid in the cell membrane. So membrane lipid are the group of the compounds which form the double-layered surface of the cells. It is also called lipid bilayer. The three major classes of the lipid which is present in the cell membrane, which is phospholipid, glycolipid, and cholesterol. Lipids are amphiphilic. They have one end that is soluble in water, that is called polar, and another end which is soluble in the fat, which is called non-polar. So this is the structure of the membrane lipid like phospholipid and glycolipid. Phospholipid contain different type of the lipids like phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. So these are commonly called phosphoglyceride. And another phospholipid, that is sphingomyelin. Next one is a glycolipid, that is different from phospholipid. So the head of the phospholipid is phosphorylated and they consist of either glycerol or sphingosine. If it contains the glycerol, hence the name phosphoglyceride, and if it contains the sphingosine, then the name is sphingomyelin and ceramide. So this is the structure of the phospholipid. Now come to the structure of the glycolipid. The head of the glycolipid contains sphingosine with one or several sugar units, which is attached to the hydrophobic chain, belongs either to fatty acids in case of phosphoglyceride or one fatty acid and the hydrocarbon tail of the sphingosine in case of sphingomyelin and glycolipid. So this is the structure of the lipid and protein in the cell membranes. So in the mitochondrial inner membrane, the ratio of protein is higher in compared to the lipid, while in the mitochondrial outer membrane, this lipid and protein is equally in equal amount and only slightly difference in the lipid, which is lower than the protein. While in muscle sarcoplasmic reticulum, the content of protein is higher in compared to the lipid. The red blood cells, the lipid content is high but lower in compared to the protein. And in the Schwann cell, the lipid content is very high in compared to the protein. So because in the Schwann cell, the lipid is responsible for making the insulating layer, that is why the content in the Schwann cell is higher. So according to the function, the ratio of the lipid is present in the membrane. So that is a different technique which we will discuss here. Now the question here, how can we measure the lateral diffusion of the lipid and protein in the membrane? So we can use the experiment that is called FRAP or fluorescence recovery after photobleaching. The first step to label the surface of the membrane with a fluorescent molecule. For example, we can label all the lipid at the surface of the membrane. Then we will expose this layer with the help of laser light that will bleach the fluorescence. So now you will see the dark spot surrounded by the sea of the fluorescent molecule, and you can measure the time it will take for this dark spot to become fluorescent again. This recovery of the fluorescence is due to the lateral diffusion of the lipids from the surrounding region into the dark spot. And you can create the graph showing how long it will take for the fluorescence to be recovered. So in this way we can measure the lateral diffusion technique in the cell membrane. So fluid mosaic model explains various observations regarding the structure and function of the cell membrane. So protein and lipid is present in the cell membrane, and apart from the protein and lipid, carbohydrate is also present. The carbohydrate is present on the outer surface of the cell membrane, which is responsible for the recognition of the cell. So there is a modification of the cell membrane. Lipid raft is one of the another modification of the cell membrane, which is mainly made up of cholesterol and sphingolipid, and it is responsible for the cell signaling, transport of the lipid, and immune function. There are a large number of the function of the lipid raft, but we will discuss here only the small portion. So this is the structure of the lipid raft, which is showing here, that is present on the cell membrane, and it can move on the cell membrane. So it can transport the lipid inside the cell membrane. We will show here the structure of the saturated fatty acid and mixture of the saturated fatty acid and unsaturated fatty acid, which is present in the cell membrane. So what will be the effect of the saturated fatty acid and unsaturated fatty acid? Because if the saturated fatty acid is present in the cell membrane, they make the strong wonderful interaction that will be responsible for the tight packing of the cell membrane. So it will provide the rigidity of the cell, while the mixture of the unsaturated fatty acid and saturated fatty acid, it provides the weak wonderful forces in the cell membrane that will responsible for the fluidity of the membrane. So here we can see there the upper structure that is showing here the paracrystalline state. When all the lipid bilayer, all the lipid molecule which have the regular or tight packing, that form the paracrystalline state, and it provides the rigidity to the cells. While the lower figure is showing that irregular packing. So irregular packing provide the fluidity to the cell membrane, and cell have low amount of the rigidity. So rigidity or fluidity can be explained by this fluid mosaic model. We can also see here the effect of the cholesterol on the fluidity. Cholesterol alter the phospholipid packaging and mobility. So if the cholesterol is not present in the cell membrane, so the cell membrane has very lower fluidity. So short unsaturated tail decrease the fluidity, while long and saturated tails, it disturb the packing and increase the fluidity. So when the cholesterol is present in the cell membrane, it will disturb the packaging and it provide the more fluidity to the cell membrane. So cholesterol is responsible for the fluidity of the cell membrane that can be explained by the fluid mosaic model. So with the help of fluid mosaic model, we can also understand the membrane asymmetry. In the cell membrane, the outer lipid bilayer is made up of phosphatidylcholine. You can see in the figure here, this is the phosphatidylcholine. So phosphatidylcholine is present on the outer leaflet, while phosphatidylethanolamine is present in the inner membrane of the cell membrane. So this is the phosphatidylcholine. It has cylindrical shape and it's present in the outer cell membrane, while this is a phosphatidylethanolamine. It is present in the inner leaflet and it has conical shape. So this presence of phosphatidylcholine and phosphatidylethanolamine provide the asymmetry of the cell membrane. Apart from the phosphatidylcholine and phosphatidylethanolamine, another molecule that is called phosphatidylserine. Phosphatidylserine is present in the inner surface of the cell membrane, and that can flip in inner to the outer surface, and such type of movement is called flip-flop movement. So flip-flop movement is another important property of the fluid mosaic model. Phosphatidylserine is a signal to the macrophage to clear the aged RBC. So many properties of the cell membrane that can be explained with the help of fluid mosaic model. So here in this diagram we can see that membrane protein. So there are three types of the protein is present on the cell membrane: lipase, flopase, and scramblase. Lipase protein is responsible for the movement of the lipid from outer surface to the inner surface, while flopase is responsible for movement of lipid from inner surface to the outer surface, and scramblase is responsible for the movement in the both direction. So in the basis of these proteins, and there are three types of the movement which are present in cell membrane. First one is a flip-flop movement. Flip-flop movement reveals the movement of the lipid by lipid molecule from one lipid bilayer to the another layer. That means this molecule will shift from inner layer to the outer layer or outer layer to the inner layer. That is, movement is called flip-flop movement. Another movement is lateral diffusion. In the lateral diffusion, the phospholipid molecule will diffuse from one position to the another position. So suppose this molecule will go here in the another, replace the another molecule. So this is the lateral diffusion. And rotational movement, all phospholipid is showing the rotational movement, that is the axial movement, or phospholipid is moving on its axis, that is a rotational movement. So three type of the movement is present in the plasma membrane that can be explained by the uh, fluid Mosaic model. Thank you.